ever stop to think about trying to see something incredibly tiny, even with like the most powerful microscopes? It's like there's this fundamental fuzziness, a limit to how sharp things can actually get.
Yeah, it's a real barrier.
Imagine trying to read, say, the fine print on a document that's just slightly out of focus no matter how much you magnify it. You just can't quite get that crystal clarity.
Exactly. And that inherent limitation, it's really held back what we can discover in biology, particularly at the smallest scales.
Right.
For a long time, scientists have been wrestling with this thing called the diffraction limit in light microscopy.
Okay.
It's basically a physical property of light itself that puts a hard cap on the amount of fine detail we can truly see.
So this blurriness makes it really challenging to understand the intricate machinery inside our cells and tissues, how molecules interact and all that.
Precisely. How tiny structures are organized, how things move around. It's all a bit fuzzy.
Okay, so let's zoom in on this diffraction limit idea for a sec. It's not just about having a dusty lens or something simple, right? This is more fundamental.
Oh, absolutely.
Yeah.
It's fundamental. When light passes through the lens of a microscope, it naturally bends and spreads out. That's diffraction. Simple physics.
Okay.
So instead of a single point of light from a tiny object showing up as a perfect pinpoint in our image, it actually appears as a little blurry disk, like a tiny splotch.
Ah, okay.
Now, the problem comes when you have two of these tiny objects really close together. Their blurry disks start to overlap.
And eventually you can't tell them apart.
Exactly. At some point, they just merge into one blur. That's the resolution limit. We literally can't resolve details finer than that blur imposed by diffraction.
So if we really want to get a handle on what's happening inside a cell, the dance of proteins, the movement of tiny compartments, we need to somehow see past this blur.
That's the goal.
And that's where super resolution microscopy techniques come in. But as amazing as they are, they often involve some trade-offs, don't they?
They absolutely do. Traditional super resolution methods have been, well, revolutionary, no question. They let us see things we simply couldn't before. However, many of them come with compromises. Some need you to blast the sample with really intense light.
Which can damage it, right? Phototoxicity.
Exactly, phototoxicity. It can actually damage or change the very thing you're trying to look at. Others take a really long time to capture just one super resolution image.
So not great for watching things that happen quickly.
Not at all. Makes it tough to study fast-moving biological events. And sometimes, getting that higher resolution means you can't image as deep or in 3 dimensions or watch processes unfold for very long. It's always a balancing act.
Right. It's like you're juggling image sharpness, capture speed, and trying to keep your sample happy and healthy all at the same time. That's a tricky equation.
It really is.
So alongside developing new hardware, scientists have also been exploring ways to use computers to sharpen those inherently blurry images you get from standard microscopes, trying to digitally de-blur the image after the fact.
Exactly. That's the whole field of computational super resolution.
Yeah.
The aim is to take those raw, diffraction-limited images and use clever algorithms to reconstruct a sharper view, revealing more of those fine details.
Okay. And our sources for this deep dive, primarily a research paper introducing a new method, they highlight a couple of main categories of these, let's say, earlier computational approaches. First, there are what they call analytical deconvolution methods. Sound a bit technical.
Yeah, it does, but the idea is fairly straightforward. Think of these methods as trying to mathematically undo the blurring caused by the microscope.
Okay, how?
Well, they use a mathematical model of how the microscope forms the image, how it blurs things, and then they try to reverse that process computationally.
Right.
One of the older and still quite common methods is called Richardson-Lucy deconvolution, or RL deconvolution.
Richardson-Lucy.
It basically works by starting with an initial guess of what the sharp image might look like. Then it simulates how that sharp image would look if the microscope blurred it, according to the model. It compares this simulated blurry image to the actual blurry image you captured. Then it adjusts its guess of the sharp image based on the difference. And it just repeats this process over and over.
Iteratively refining the guess.
Exactly. Iteratively tweaking it until, hopefully, the simulated blur matches the real blur and the guess represents a sharper underlying reality.
Okay, so it's like digitally nudging an out-of-focus picture back towards sharpness using a model of the blur. Makes sense. And the sources also mention sparse deconvolution. What's the thinking there?
Sparse deconvolution works on a slightly different assumption. It assumes that many biological structures, especially at the nanoscale, are actually quite sparse.
Sparse meaning?
Meaning that Only a small number of points or lines are actually bright against a mostly dark background. Think individual molecules or maybe thin filaments.
Okay, yeah. Lots of empty space.
Right, so the algorithm then tries to find the simplest, most sparse, sharp image, the one with the fewest bright spots or lines, that when you apply the microscope's known blurring effect, would result in the blurry image you actually observed.
So it's looking for the most minimal explanation for the blurry data, assuming the underlying reality is sparse.
You got it. Find the fewest possible bright spots that could create that blurry pattern.
That makes intuitive sense too. But these analytical methods, they have their downsides, right? Our sources point out some limitations.
Oh, absolutely. A big one is that they often need quite a bit of manual fiddling.
Parameter tuning.
Yeah, the user needs to adjust various settings within the algorithm, often based on their own experience and the specific data set. And the final results can be pretty sensitive to getting those settings just right.
So it takes expertise and can be subjective.
It can be. Plus, they can be computationally quite heavy and slow, especially for large 3D data sets. You might be waiting hours or even longer. And maybe the biggest challenge is noise. They often struggle when the original images are noisy, low signal-to-noise ratio or low SNR.
What happens then?
Well, these methods can sometimes end up amplifying the noise along with the signal. Worse, they can sometimes even create artificial details, little blobs or lines in the final image that weren't actually in the sample at all. Artifacts.
Right, so you start with blurry and noisy, and you might end up with sharper, but still noisy, maybe with some fake stuff thrown in. Not ideal.
Definitely not ideal.
Okay, so what about the other major category our sources discuss? That's supervised deep learning for super resolution, or DLSR. Now, deep learning has exploded everywhere, so it must have impacted microscopy imaging too.
Oh, tremendously. Supervised deep learning methods use these powerful artificial neural networks. You train them by showing them vast amounts of paired data.
Paired data, meaning?
Meaning you need lots of examples where you have both a blurry, low-resolution image and its corresponding perfectly sharp, high-resolution version, the ground truth.
Ah, okay. Like a before and after pair.
Exactly. The network learns the complex mapping, the relationship between the blurry input and the sharp output, by seeing thousands, maybe millions, of these example pairs.
And once it's trained?
Once it's trained well, these networks can be incredibly effective. They can often produce really impressive super-resolution results on new blurry images, and crucially, they can do it very, very quickly, much faster than the iterative analytical methods.
That sounds like a huge leap. Speed and potentially better quality.
So, what's the catch? Why isn't supervised deep learning the answer to everything here?
The catch is that word supervised. It all hinges on having access to those large, high-quality paired data sets.
The blurry image and its perfect sharp counterpart.
Right. And in many real-world biological imaging scenarios, especially when you're trying to study living cells or dynamic processes, getting that perfectly matched ground truth data is incredibly difficult. Sometimes it's just impossible.
Why is that? Well, think about it. How do you capture a truly high-resolution, artifact-free snapshot of, say, a molecule that's constantly moving inside a living cell? At the exact same moment, you capture your blurry, lower-resolution image needed for enhancement.
Yeah, I see the problem. The high-res methods might need different conditions, or more light, or take longer.
Exactly. Or the very act of getting the high-res image might damage the sample you're trying to observe non-invasively. It's a real chicken-and-egg problem. You need the sharp image to train the network to see sharply, but you often can't get the sharp image of the thing you want to study sharply.
Oh, that's a great way to put it. So while supervised methods are powerful tools if you have the right data, they're often not practical for pushing the frontiers in live cell imaging or other tricky biological contexts.
That's the major bottleneck, yes.
Okay, so this brings us squarely to the focus of our deep dive today. A newer approach called the zero-shot deconvolution network, or ZS-DeconvNet. The name itself, zero shot, sounds like it might tackle this data problem head-on. What exactly is it?
It is. ZS-DeconvNet's also a computational super-resolution method using deep learning. But its core innovation, the really big deal, is that it's a zero-shot or unsupervised deep learning method.
Zero shot, unsupervised. Okay, unpack that for us. What does that mean in practice for enhancing microscope images?
It means ZS-DeconvNet does not need those large collections of matched, blurry, and sharp images for training. It doesn't need the ground truth.
Wow. So how does it learn them?
It learns directly from the noisy, low-resolution data that you actually acquire with your microscope. You can even learn from just a single blurry image, or maybe a short time series, or a 3D stack of
From
images.
the blurry image itself, without ever seeing a short version.
That's the key. This ability to learn directly from the data at hand without needing pre-existing perfect examples is what makes it so potentially transformative, especially for those difficult biological applications.
Okay, that sounds almost like magic. How does it possibly manage to do that? How does it know what sharp looks like if it only sees blurry?
It's not magic. It's actually based on a very clever principle that uses our understanding of the microscope itself. It leverages what's called an optical imaging forward model.
A forward model, meaning a model of how the image is formed.
Exactly. A mathematical description of how the microscope takes a theoretical perfect sharp image and turns it into the blurry, noisy image you actually record.
Hmm.
So the network takes your noisy input image and tries to produce a high-resolution output guess.
Okay.
Then, and this is the crucial bit, it uses that forward model to simulate what would happen if that high-resolution guess were put back through the microscope. It simulates the blurring and downsampling process.
Ah, so it predicts what the blurry version of its own sharp guess would look like.
Precisely. And the network is then trained to make that simulated blurry image match the original noisy, blurry input image as closely as possible.
I think I'm getting it. So instead of comparing its output to a known sharp image, it compares a degraded version of its output back to the original input it started with.
You've got it. It's learning the inverse of the image degradation process by essentially trying to perfectly reconstruct the input, but by way of guessing a higher resolution version first. It's a self-consistency check.
That is a really elegant idea, learning to sharpen by learning how to accurately re-blur. But hang on, what about the noise? Microscope images are always noisy. If it's just trying to match the noisy input, wouldn't it risk learning to reproduce the noise as well, thinking it's part of the real structure?
That's a fantastic question and a critical challenge they had to solve. And they came up with really ingenious ways to handle it, slightly differently for 2D and 3D images.
Okay, tell me about the 2D version first. How does it deal with noise in a single image?
For the 2D implementation, they use something called an image recorruption scheme. It sounds complex, but the idea is this. You take your single noisy input image, then you computationally generate two slightly different, independent, noisy versions of it.
Two versions from one. How?
They use statistical models of the noise, typically found in microscopy, usually a mix of Poisson and Gaussian noise. They essentially simulate adding different random noise patterns that follow those statistics to the underlying signal inferred from the input image. So you end up with the two images that represent the same underlying biological structure, but each has its own unique, statistically independent noise overlay.
Okay, so two unique noisy renderings of the same scene. Then what?
Then during training, these two recorrupted images act as the input and the target pair for the network. The network tries to predict one noisy version from the other noisy version.
Ah, so because the underlying signal is the same in both, but the noise is different and random.
The network learns to identify and preserve the consistent part, the actual biological signal, while learning to effectively ignore or filter out the inconsistent part, the random noise. It learns the structure, not the static.
That is brilliant, using self-generated noisy pairs to teach itself denoising. Okay, so what about the 3D version? How does it handle noise in a volume stack without this recorruption step?
For 3D data, they use a different but equally clever technique called spatially interleaved self-supervised learning.
Spatially interleaved.
Okay, think of your 3D stack of images like slices through the sample at different depths, right? This method essentially divides the stack into, say, the odd-numbered slices, slice 1, 3, 5, etc., and the even-numbered slices, slice 2, 4, 6, etc., along the depth axis, the z-axis.
Okay, splitting the stack.
Then, the network is trained to predict the even-numbered slices using only the odd-numbered slices as input, and vice versa.
Why does that work for denoising?
Because adjacent slices in a typical 3D microscopy stack are usually very similar in terms of the underlying biological structure. They show almost the same thing. But importantly, the noise pattern in each slice is largely independent and random.
Ah, I see. So again, it's learning to predict something very similar, the adjacent slice, from an input that has different random noise.
Exactly. It learns to extract this shared structural information while suppressing the uncorrelated noise between adjacent slices. They also add something called a gap-amending regularization, or GAR term, which helps handle any subtle, real differences or misalignments between the odd and even slices, making the prediction more robust.
So it's using the inherent redundancy and noise independence in the 3D data itself as its own teacher for denoising. Very clever. Now, our sources also mention a dual-stage network architecture. Why split the network into two parts?
Good question. ZS-DeconvNet typically uses a deep learning network structure that's divided into two main stages. Stage 1 acts primarily as a denoiser. Its job is to take the initial noisy input and clean it up as much as possible.
Okay, noise reduction first.
Then the output of this denoising stage, a cleaner but maybe still blurry image, is fed into the second stage. This second stage focuses specifically on the deconvolution part, sharpening the image and enhancing the resolution.
So denoise first, then deconvolve. Why is that better than doing it all at once?
It's used to help stabilize the training process and often leads to better results. Trying to simultaneously learn how to denoise and deconvolve perfectly can be very challenging for a network. Separating the tasks seems to make each stage more effective and can help prevent the network from introducing noise-related artifacts during the deconvolution step.
Makes sense. Divide and conquer. And they use specific types of networks for these stages.
Yeah, for 2D images, they often use a type of network called a UNet, which is known to be very good at capturing both fine details and broader context in images. For 3D data, they might use architectures like RCNs, residual channel attention networks, which have shown strong performance in various image restoration tasks, including super resolution.
Okay. And there's one more technical bit mentioned, Hessian regularization. That sounds pretty math-heavy. What's its role?
Yeah, the name sounds intense, but think of Hessian regularization as a kind of quality control or guidance mechanism for the network. It helps to ensure that the final reconstructed image looks natural or biologically plausible.
How does it do that?
It essentially penalizes solutions that have overly sharp, unnatural edges or textures that look more like noise patterns than smooth biological structures. It encourages smoothness in areas that ought to be smooth, while still allowing for sharp, well-defined features where they should exist. So it sort of nudges the network away from generating weird artifacts and towards producing a cleaner, more realistic-looking result. It regulates the convergence.
Like a smoothness prior, almost. Discouraging jaggedness.
Kind of, yeah. It helps mitigate reconstruction artifacts and keeps the network's output looking sensible.
Got it. And finally, perhaps most critically, it sounds like the point spread function, or PSF, of the microscope is essential for ZS-DeconvNet to work. Can you just remind us what the PSF is and why it's so crucial here?
Absolutely. The point spread function, the PSF, is basically the fingerprint of your specific microscope's optical system. It describes exactly how that system sees an infinitesimally small point source of light.
So it's the blur pattern for a single point.
Precisely. Instead of a perfect point in the image, you get this characteristic blurry pattern defined by the PSF. Now, ZS-DeconvNet explicitly incorporates the measured or calculated PSF of the specific microscope used to get the data into its deconvolution process, particularly in that second stage.
Why is that so important?
Because the network needs to know the exact nature of the blur it's trying to undo. The PSF tells it precisely how the microscope blurred the original sharp image. By having this accurate information, the network can much more effectively estimate the underlying high-resolution structure that must have produced the blurry image it sees.
So knowing the specific blur is the key to reversing it accurately.
It's absolutely critical for accurate deconvolution, yes.
Yeah.
It's like having the specific decryption key for your microscope's blurring effect.
Okay, wow. So putting it all together, ZS-DeconvNet is this really sophisticated, unsupervised deep learning method. It learns to deblur and denoise microscope images by using a model of the microscope's blurring, the PSF, and clever self-supervision tricks using the noisy input data itself without needing any separate sharp ground truth images.
That's a great summary.
That sounds like a massive leap forward. What are the really key advantages, the breakthroughs that this approach unlocks for scientists?
Well, the zero-shot unsupervised nature is, as we've discussed, probably the biggest advantage. It completely sidesteps the need for paired training data, which is huge for studying live, dynamic, and especially light-sensitive biological samples. That opens up so many experiments that were previously difficult or impossible.
Right, tackling the data bottleneck.
Exactly. And related to that is its incredible data efficiency. It can genuinely be trained on just a single noisy image or a 3D stack. Compare that to supervised methods needing potentially thousands of pairs.
Huge difference. What about performance compared to the older methods?
When compared to traditional analytical methods like Richardson-Lucy or sparse deconvolution, the studies show ZS-DeconvNet generally achieves significantly better resolution enhancement and overall image quality things measured by metrics like PSNR, peak signal-to-noise ratio.
So sharper and cleaner images.
Yes. And crucially, once the network is trained, which might take some time initially, depending on the data size, applying it to new images is incredibly fast. We're talking processing times on the order of milliseconds per image.
Milliseconds, compared to potentially hours for sparse deconvolution.
Exactly. That speed difference is absolutely transformative for things like real-time analysis during live cell imaging experiments or processing the massive data sets generated by modern microscopes.
That speed must enable researchers to see things happening almost as they occur, but with super resolution clarity.
That's the potential, yes. Plus, because it learns directly from the input data, ZS-DeconvNet is inherently adaptable. It adapts to the specific imaging conditions, the unique noise characteristics of that dataset, and the type of sample being imaged without needing a perfectly matched pre-existing training set.
So it tailors itself to this specific experiment.
In essence, yes. Which also means it offers the potential to instantly enhance the capabilities of existing microscopes. You don't necessarily need new, expensive hardware. You could potentially get better results from the microscope you already have just by applying this smarter processing.
That's a very appealing prospect for labs.
It really is. And by enabling super resolution from images that can be taken with lower light levels and faster acquisition speeds, because it's good at handling noise and extracting signal, it effectively boosts what researchers call the spatiotemporal bandwidth of microscopy.
Meaning you can see finer details in both space and time simultaneously.
Exactly. You overcome some of those trade-offs we talked about earlier. The paper demonstrates achieving more than a 1.5-fold improvement in resolution beyond the theoretical diffraction limit.
1.5-fold is significant.
It is.
Yeah.
And maybe even more impressively, they showed it can work effectively even when the fluorescence signals are 10 times weaker than what's typically required for good super-resolution imaging.
Wow. 10 times less light. That must be huge for sensitive live samples.
It's absolutely critical for observing delicate biological processes over extended periods without causing significant phototoxicity or photobleaching. Being able to image longer and gentler is often key to understanding biology.
10 times less light. That really is a monumental step. And it sounds like this technique isn't just limited to one type of microscope either. Our sources say it's quite versatile.
Yes, that's another major strength. Its broad applicability.
Right.
The researchers have successfully demonstrated ZS-DeconvNet working well with a whole range of important microscopy modalities,
Such as?
including turf microscopy, which looks at surfaces, 3D wide-field microscopy for basic volume imaging, confocal microscopy, which gives optical sections, two-photon microscopy for imaging deeper into tissues, lattice light sheet microscopy, known for being very gentle on samples, and even several types of structured illumination microscopy, or SIM, like turf SIM, 3D SIM, and lattice light sheet SIM.
That's a really diverse list. It covers a lot of the workhorses in biology labs.
It really does. This wide range shows its potential to improve imaging across a huge spectrum of biological research, from single molecules to tissues.
Okay, but it's not just about getting prettier pictures, right? The real test is whether this technology actually enables scientists to make new discoveries, to see biological processes they couldn't see clearly before. Can you share some specific examples from the paper where ZS-DeconvNet made a real difference?
Absolutely. That's the crucial part, the biological impact. The paper highlights several really compelling examples. For instance, they used it to perform long-term, two-color super-resolution imaging of how F-actin and myosin tech interact during cell adhesion and migration.
Actin and myosin, key players in the cell's skeleton and movement.
Exactly. These processes are very dynamic and highly sensitive to light damage. Using ZS-DeconvNet allowed them to observe the fine details of how the cytoskeleton reorganizes and how the myosin motors generate force over extended periods in living cells without causing significant harm.
And what did they learn?
They gained new insights into the mechanics of how cells crawl, particularly the role of different myosin II structures in that process. Details that were previously obscured by the resolution limits or the inability to image long enough due to phototoxicity.
That's fantastic. Watching those fundamental processes unfold live at that resolution, that must provide so much new information. What other biological systems did they look at?
Another really neat application was visualizing the dynamics of the endolysosomal system. These are tiny vesicles inside cells involved in trafficking, sorting, and degrading molecules.
Like the cell's postal and recycling service?
Kind of, yeah. And they move incredibly fast. By applying ZS-DeconvNet to images where the proteins were expressed at their natural endogenous levels, which is important to avoid artifacts from overexpression, they could track these tiny vesicles.
Well, could they?
They could track recycling endosomes and late endosomes zipping around the cell over long distances, observe them fusing together or splitting apart, fission and tethering events, all with unprecedented clarity and over long time courses. Seeing these rapid dynamics at endogenous levels is key to understanding normal cellular function.
It must be like watching rush hour traffic inside the cell, but finally being able to see the individual cars and how they interact. And was it just limited to single cells on a dish?
No, they push it further. They demonstrated long-term 3D super-resolution imaging of multiple organelles simultaneously, things like the ER, mitochondria, chromosomes, nuclear speckles, all during mitosis, the process of cell division.
Mitosis is incredibly complex and dynamic, and also very light sensitive, right?
Exactly. It's a huge challenge to image well. But they were able to observe the intricate choreography of these organelles reorganizing throughout the entire process over thousands of time points in 3D in sensitive cells. That's a major technical achievement.
Wow, thousands of time points in 3D super resolution during mitosis.
And beyond single cells, they also successfully applied ZS-DeconvNet to image larger multicellular specimens. They showed high-resolution 3D images of immunostained mouse embryos and also developing C. elegans embryos.
So applications in developmental biology, too.
Definitely. It highlights the potential for getting much finer structural details in complex developing organisms, which could be huge for understanding how tissues and organs form.
It really sounds like it could revolutionize how we study life from the subcellular level right up to developing organisms. The paper also mentioned using it to improve SIM images even further. Structured elimination microscopy.
Yes, that's right. They developed a version called ZS-DeconvNet SIM. SIM is already a super-resolution technique, but applying ZS-DeconvNet on top of the standard SIM reconstruction process can push the resolution even further and significantly improve the signal-to-noise ratio.
So it cleans up and sharpens SIM data.
Exactly. They showed examples where ZS-DeconvNet SIM could resolve very fine structures, like the details of clathrin-coated pits involved in endocytosis or the intricate meshwork of the cytoskeleton much more clearly than conventional SIM alone. For lattice light sheet SIM, they even showed it helped achieve nearly isotropic resolution, meaning the resolution became almost equally good in all three dimensions, X, Y, and Z, which is often a big challenge in 3D microscopy.
So it's not just an alternative to other methods, it can actually enhance other cutting-edge techniques. That's really impressive adaptability. Now, for any scientific tool, accessibility is key for it to have a broad impact. Is ZS-DeconvNet something only deep learning experts can use, or have they made it usable for, say, a typical biologist?
That's a crucial point, and they recognized it. The researchers have actually put significant effort into making it accessible. They developed a user-friendly plugin for Fiji.
Fiji. Oh, yeah, that's the image processing software almost every biologist uses, right? ImageJ's distribution.
Exactly. It's incredibly popular and widely used in the life sciences community. This plugin essentially packages the power of ZS-DeconvNet in a way that researchers who aren't deep learning specialists can easily use it.
So a biologist could potentially take their own microscope data, open it in Fiji, and use the plugin.
Pretty much. With the plugin, users can train a ZS-DeconvNet model specifically on their own data, which is important for getting the best results, and then apply that trained model to enhance their images, often with just a few clicks within that familiar Fiji environment.
That's fantastic. Lowering the barrier to entry like that is absolutely vital for widespread adoption and allowing more labs to benefit from this technology.
Agreed. It makes a huge difference in how quickly and broadly a new computational tool gets used.
Okay, so stepping back now and thinking bigger picture. The core idea behind ZS-DeconvNet, using a forward model of the measurement process to computationally extract clear information from noisy data without needing perfect ground truth examples, that seems like a powerful concept. Could it be applied in other scientific fields beyond just microscopes?
Oh, absolutely. That's a really interesting direction to think about. The fundamental problem trying to get the best possible information from imperfect, noisy measurements when you know something about how those imperfections arise is incredibly common across science.
Like where else?
Well, consider astronomy. Telescopes, whether ground-based or in space, capture light from distant objects. But those images are always blurred by the atmosphere, by imperfections in the telescope optics, and affected by detector noise.
Same problem, different scale. Blur and noise.
Exactly. So a similar approach using a well-characterized forward model of the telescope and atmospheric blurring, like the PSF concept, within an unsupervised or self-supervised learning framework, could potentially be used to deblur and enhance astronomical images, maybe allowing us to see fainter galaxies or finer details on planets with greater clarity.
That's a fascinating parallel. Seeing the incredibly tiny in biology, seeing the incredibly distant in astronomy, similar computational challenges. Any other areas spring to mind?
Medical imaging is another obvious candidate. Think about MRI or CT scans. There's often a trade-off between image resolution or quality and things like scan time or, particularly for CT, the radiation dose given to the patient.
Right. You want the best image with the lowest dose or fastest scan.
Precisely. So could an unsupervised deep learning approach, informed by the physics of MRI signal generation or X-ray attenuation, allow us to reconstruct high-quality, high-resolution images from data acquired using lower radiation doses or significantly faster scan protocols? That would be a huge benefit for patients.
Yeah, safer and more comfortable scans with potentially better diagnostic information. That would be impactful.
Definitely. And you could even think about fields like audio processing. Imagine trying to denoise old, noisy audio recordings where you don't have a clean version. If you have some model of the noise characteristics or the recording process, maybe a similar self-supervised approach could help restore them.
So the key ingredients are having noisy data, needing better resolution or less noise, and having a reasonably good understanding, a forward model of how the clean signal got degraded into the noisy data you have.
That's the core recipe, yes. If you have those elements, then this kind of zero-shot, model-informed, self-supervised learning approach holds a lot of promise across many different domains.
It's really exciting to think about the potential ripple effects of these computational ideas. Of course, as with any technology, ZS-DeconvNet isn't perfect. What are some of its current limitations, and where might future research go?
Right, it's important to be aware of the limitations. The researchers themselves point out a few. For example, if you have extremely weak fluorescence signals that are barely distinguishable from the background noise, ZS-DeconvNet might sometimes struggle to recover them accurately. It might mistake that very faint signal for noise and suppress it.
Okay, so a sensitivity limit at the very low end.
Potentially, yes. Also, while it adapts to the training data, a model trained on one very specific type of data or microscope setting might not perform quite as well if you apply it to data that looks significantly different, say, a different cell type, different staining, or a different microscope altogether. There can be generalization issues.
So retraining on the specific data type is often still recommended.
It generally gives the best results, yes. And as we discussed, the accuracy of the point spread function, PSF, used during training, is really critical. If the PSF model doesn't accurately match the real PSF of the microscope used for the experiment, the deconvolution quality can suffer. Getting an accurate PSF can sometimes be tricky.
Those seem like practical challenges. What about future directions for improvement?
There are many exciting avenues. Researchers could integrate the ZS-DeconvNet principles with even more advanced, newer deep learning architectures that might be even better at capturing fine details or handling complex noise. There's also huge potential in applying this zero-shot learning philosophy to enhance other types of super-resolution data, like from techniques such as Palm, Storm, or STED microscopy.
Extending the unsupervised approach to other SR methods.
Exactly. Another area is improving generalization developing techniques, perhaps borrowing from domain adaptation research that would allow a model trained on one data set to work better on different ones without needing full retraining. And a really significant advance would be expanding the method to handle situations where the PSF isn't the same across the whole image, a spatially varying PSF. This happens in techniques like light field microscopy or when using adaptive optics to correct aberrations. That's a much harder problem, but would broaden the applicability even further.
So plenty of exciting research still to be done to refine and extend this powerful approach.
Okay, well, this has been a really enlightening deep dive into ZS-DeconvNet and the landscape of computational super-resolution. To quickly recap the key takeaways for everyone listening, it sounds like ZS-DeconvNet represents a really significant step forward. It tackles a major bottleneck in microscopy, the need for paired training data, by using this clever, unsupervised, model-informed, deep learning approach.
That's right. It makes high-quality computational super-resolution much more practical, especially for those challenging live cell, long-term, or low-light imaging experiments where getting ground truth data was prohibitive.
And its adaptability across many different microscope types, its impressive data efficiency, and crucially, the effort to make it accessible through tools like the Fiji plugin, all point to its potential for real, broad impact in biological research. Definitely. By essentially allowing researchers to see more detail over longer times with less light, using potentially the instruments they already have, it's poised to help us uncover biological details and dynamics that were previously hidden. It really could lead to a deeper, more nuanced understanding of how life works at the cellular and molecular level.
It really makes you stop and think, doesn't it, about the power we unlock when we combine our sophisticated measurement tools, like microscopes, with equally sophisticated computational tools that can intelligently interpret the data. What new questions can we start asking? What new discoveries are waiting when we can effectively upgrade our vision just by processing the data more cleverly?
It opens up exciting possibilities.
We really encourage you, if this has piqued your interest, to explore further. Check out the original research paper on ZS-DeconvNet. We'll put a link in the show notes. Maybe explore the Fiji plugin or the tutorial homepage if you work with microscopy data.
Yeah, see if it could be useful for your own work.
It's a fantastic example of how computational innovation is driving progress across science, and it leaves you wondering, what other fields are poised for similar breakthroughs by applying these kinds of model-aware, data-driven approaches to extract more from the measurements we can make? Really exciting times ahead.